Let's go through the talk. It'll probably be about 30 minutes, and then there'll be plenty of time for questions. My co-author and I put together this idea, and we had a hard time doing it, in large part because we haven't really found any alien life to study. So a lot of it is based on the idea that we understand life on Earth, and we can extrapolate. And I think that leads us to some conclusions that are relevant to those of us interested in colonizing other planets. One term I want to get into before we get too far is that of the biosphere. And the biosphere is that part of a planet which contains life. Here on Earth, you can go down several miles and you'll find bacteria living in the crust, and it's very happy there. Go down 100 miles and you probably won't find much. Certainly the surface of the planet is covered with life. Oceans are covered with life. And if you go up several miles into the atmosphere, again, you're going to find animals and bacteria. So that's the definition of the biosphere. It's where life exists on a planet and how that life interacts with itself. And the reason for putting together a talk like this is the perception that colonizing other planets is a two-part problem. First, you find a habitable planet, and then you figure out how to get there. And when we say a habitable planet, we're talking about a second Earth, just like this one. It's going to have, Earth normal gravity, oceans. If it's a habitable planet, it's got to have oceans. It's going to have an atmosphere very similar to what we have here. That means, probably green plants, hopefully friendly natives, tasty animals, and in other words, just move-in ready. You figure out how to get there, and then you can land, and life is good. I think part of that is due to the idea or the concept from Star Trek called M-class planets. Who is not familiar with M-class planets from Star Trek? Okay, so several, all right, not too many. In Star Trek, Captain Kirk and friends were always running into M-class planets. And what that is a planet, very much like Earth. People can breathe the air, the temperatures are not too bad, and it's real good for film crews, and you don't have to dress up in spacesuits. But it led to the narrative that there's a lot of planets like that out there, and it might be. Part of this talk, we're going to have to get back into the evolution of life. And, let's assume that we have a planet with conditions very similar to our early Earth. On Earth, life first appeared about 4 billion years ago. In other words, as soon as conditions were somewhat favorable. And so, based on that one data point, I guess we can assume that life is going to be fairly common. As soon as conditions are somewhat favorable, you're going to have life, and then it's going to evolve over some period of time in some directions. So life appears, it evolves, and we're going to run into it. Oh, let's see, oxygen. A lot of science fiction stories, they'll be wandering along and they'll find a planet with oxygen in there and there's no life. But if there's oxygen in the atmosphere, that is an indicator that you have life on the planet. Oxygen is a waste product of photosynthesis. Again, this ability developed in plants here on Earth about 3 billion years ago. So it took a while to get around to this, but it's a really neat trick. The bacteria, the algae, takes CO2 from the atmosphere, it takes water from the environment, takes light from the sun, and it makes carbohydrates, which is a really neat way to store energy for the plant. And then animals that eat plants also, get that energy. But one of the waste products from that, of course, is oxygen. So if you have enough algae or bacteria doing this over a long period of time, you end up with what we have here, which is an atmosphere of about 21% oxygen. Now, if all the plants were to die today, the oxygen would still be there, but it wouldn't be there for very long. Came across a couple of studies that said if the plants disappear, it'll take several thousand years, but then the oxygen disappears. And we end up with an atmosphere of nitrogen and CO2. So if there's life, there's oxygen, or if there's oxygen, there's life. And if we have alien life, suppose we find our Earth 2, it's a nice planet circling a yellow star, got good gravity, it's got oxygen, nitrogen atmosphere. And again, remember, if there's oxygen, then there's life in there, or on the planet somewhere that's producing this stuff as a waste product. That means probably green plants, insects, animals, oceans, and so on. Assume we can get a colony ship there. That's not a minor problem either. But assume we get there, then the question is, can we move into our new home, Earth 2? And I would submit that there's two considerations. One is ethical. Can we move into someone else's home? And I'm going to very carefully avoid that question. And the second one is there a compatibility problem of us moving into someone else's planet? Again, we're jumping around a little bit here, but there's a lot of threads that need to be pulled together. Building blocks here on Earth are the amino acids. Those are the basic building blocks for all life here on Earth. The Miller-Urey experiment, I guess hopefully everyone here is familiar with that. They took some simple inorganic chemicals and water, ran it around some apparatus, had a spark in there. And over time, they developed reasonably complex organic chemicals consisting of, in some part, the amino acids. There are about 300 naturally occurring amino acids, and if you engineer the molecule, you can probably make thousands more. So one of the assumptions I'm making is that if we run into alien life on a planet, similar to ours, it's going to use the amino acids as a building block, just like us. And amino acids are very useful, in part because of their ability to form peptide bonds. If you take two amino acids and you hold them just right, and you bang them together with just the right amount of energy, a couple of things happen. These atoms over here get together, form a water molecule, and wander off. The remnants create a peptide bond, and two molecules become one. They get together, form water, wander off, and you have a peptide bond. Peptide bond is actually pretty strong. It's not going to break easily, but it can be broken. And you can do this again and again and again, and you end up with a polypeptide chain, with each one of these being an amino acid. And it's called a polypeptide chain. If it's over 50 amino acids in length, it's generally called a protein. And proteins are the amino acids. You've heard of a carbon-based life form. Well, there's the carbon. It's attached to an amino group here, an acid group here, a hydrogen here, and then a variable R group, depending on what the function of the amino acid is. And amino acids can come in right-handed or left-handed versions. For some reason, Earth life has decided to standardize on left-handed versions. If you make these things in a test tube in a lab, you're going to get roughly 50-50. But life, for some reason, has settled on left-handed versions of these molecules. And these are the 20 amino acids that make up us, and worms, and cats, and dogs, and bacteria. And what is in blue is the R group. And as you can see, it varies quite a bit. This is not intended to be a biochemical lecture, just get a flavor of what the amino acids look like. And here's another way to look at the amino acids. These 20 here are the same ones we saw before. The ones in green are known as essential amino acids. They're essential because we can't make them in our body. We have to obtain them by ingesting food, plants, animals. They're composed of proteins. Our digestive systems take these proteins and they actually break the protein down into its constituent amino acids. That's released into the bloodstream, and then the bloodstream takes this around to all of our cells as part of the nutrient supply that it provides. So, essential amino acids. Now these ones over here we can make, but we can also obtain from plants and animals. And notice that there are 3 odd ones over here. This one is essentially the 21st amino acid that humans need, and we'll talk about that in a little bit. And these 2 over here aren't used by humans, they're used by bacteria and arachnids, of all things. So here's the collection of all the building blocks that life on Earth is made of.
There were 24. There were 3 rows of 7 and 1 row of 3, but it said it was labeled 23. So.
Yes. But this is just a legend. So that's not really an amino acid. So okay, trick question, but you caught it. Very good. Proteins, they're vital for life. That's what membranes and structures inside the cell are made of. They're used to construct nanomachines that operate inside the cell. They serve as messengers between cells. They're hormones. They're important to the creation of biologically important molecules. The body can break them down and use them for energy, or the body can break down proteins and use them as amino acids that they then use to make other proteins. And the enzymes within the cell are in large part proteins. So this is who we are, proteins. The 21st amino acid, most amino acids are pretty stable. This one is very reactive. And rather than floating around waiting to be used, it's essentially made on demand inside the cell. And what it is, it's a modification of the amino acid serine. And what we're doing, I need more hands here. Here's a selenium atom right here. And here's serine. What we're doing is replacing an oxygen with a selenium atom. And there's 2 or 3 enzymes that are important in this process. And again, this amino acid is made pretty much to order for the cell. It's used in over 50 proteins, so it's very important. Turns out that lack of selenium results in a number of diseases where we are unable to make this amino acid, and that results in sometimes death. Now in East Tennessee, we're in pretty good shape. There's lots of selenium in the soil, so we pick it up on our food. But there's areas in China where they're lacking it, and they've run into deficiencies related to this. So 21 amino acids for people, and then arachnids have another one, and there's yet one more, I guess, for bacteria. Ribosomes. If you don't believe nanotechnology is possible, look inside the cell. This is a really neat machine. It is in essence a protein factory. It's located in the cell, but outside the nucleus. It reads a messenger RNA, which is created in the nucleus, floats out, the ribosome grabs it, and reads it much like a punch tape. And this provides the information telling it how to build a protein. And so based on the information here, it'll grab a particular amino acid and slam them together, making a peptide bond. And thus the polypeptide chain or protein emerges here. Notice that the messenger RNA is read, but it's not destroyed, so it can be used over and over and over. This is a pretty big machine. The molecular weight is 4.2 million. The molecular weight of hydrogen is 1, to give you some idea of how big this thing is. It's composed of 80 polypeptides / proteins, and 4 strands of RNA that actually come from the nucleus. And all these things are assembled into a machine that once it's assembled is actually very stable and can generate proteins for many years. So life is pretty remarkable.
You say 70 or 80 years, is that what you're getting at?
No, I didn't say that. I didn't say that. The books I read didn't have any idea. My co-author didn't have any idea. Some, probably go back to birth, but, active cells have 1 million of these things. Inactive cells have far fewer. So there's a lot of them. Some of them are probably pretty old. Here's another artist impression of a ribosome. This is the messenger RNA being read and read and dispelled. Here's the protein that's emerging from it. And this thing can click along pretty quickly. Again, the books I looked at said that these things can assemble the amino acids at the rate of 4 or 5 a second. So, it clicks right along. Of course, it's on a very small scale, but it clicks right along. But what happens when the messenger RNA calls for a particular amino acid that's not available? The process stops. And somehow it signals to the rest of the cell, hey, I need this. And it then begins to break down other proteins, hoping to free up the amino acid that this thing needs to continue its work. And usually 2 or 3 proteins are destroyed for every one created. This is sometimes known as malnutrition. So alien biospheres, let's assume that the aliens use amino acids, assume that they evolved along a different pathway, similar but different. Weber and Miller, 2 researchers, put out an interesting paper where they looked at the amino acids that are available in the primeval, soup of the earth, and concluded that, based on simplicity and ease of manufacturing the things, that an alien life form evolving under those conditions would probably use about 75% of the amino acids that make us us. But that means 25% would be different, would be alien to us. And this is, an estimate, probability. Could be more, could be less. And also the number 20 or 21 is not special in any way. They might actually use more amino acids. If that's the case, then their cellular machinery is going to have to be a lot more complex, but the resulting proteins could be more specialized and perhaps more efficient. So if you're looking for a superior race, maybe they just have more amino acids. So carbon-based aliens, here we go. If we run into them, if we're running around an alien biosphere, suppose they have lots of fruit and animals, that we can eat. In all likelihood, or there's a possibility, perhaps a good possibility, that they won't have all of the essential amino acids that we need. And if you don't have the amino acids, if it's essential, we can't make it and we die pretty quick. There's also a 50-50 chance that they're going to standardize on a different handed version of the amino acids. I said that life has standardized on left-handed amino acids. If they standardize on right-handed amino acids or some mixture, we won't be able to use any of those amino acids to construct proteins. It's like having the wrong glove. Left-handed glove won't fit on the right hand. Just doesn't work. So the conclusion is, if you're a human running around on an alien world, trying to exist in their biosphere, in all likelihood, you're going to, suffer malnutrition and probably die. A couple of ways to get around that: nutritional supplements. You can bring food with you. Maybe you can plant earth food on the alien world and then you exist off that. Or you can modify people to be consistent or compatible with the alien world. But it's not just a matter of malnutrition. It could be something worse. One example is the amino acid muscazone. Muscazone is an amino acid produced by some mushrooms. It turns out to be a toxic psychoactive chemical. The mushrooms don't use it for anything except self-defense. It discourages animals like us from eating them, so it is pretty effective in that arena. But if you eat a couple of mushrooms, you're getting microgram quantities. But imagine if the alien life form is using this as one of their standard amino acids. Then instead of getting microgram quantities, if you had an alien steak, you're getting gram quantities of a toxic psychoactive material. And this looks a lot like, some of the other amino acids, that I showed earlier. It's not one that we use, but it might be one they use. Another problem is analogs. This is the analog of lysine. Lysine looks just like this, except this is a carbon. So it's very similar to lysine. This is a man-made amino acid developed here fairly recently. They think it has uses in drugs and other areas. But what they've noticed is if you grow cells in the presence of this amino acid, the cells are not as viable as normal cells. And what we think is happening is the ribosome has never run into this before. It can't tell the difference. So sometimes it grabs lysine, sometimes it grabs the analog, but they're not the same. The protein will not function as well, may not function at all, and so therefore the decreased viability. And all of the amino acids that we have analogs that the ribosome may not be able to differentiate. Pheromones and pollen, here on Earth they're very useful. Pheromones for communication between organisms, and pollen as a way for plants to transmit genetic information back and forth. But on an alien world, they're probably going to develop the same abilities, but their pheromones and pollens are going to be alien, and perhaps, maybe it's going to be harmless to humans, maybe it's going to be similar to ragweed, maybe it's going to be ragweed squared. Perhaps it's psychoactive. We just don't know. And if there are any writers out there, just imagine if it's a toxic nerve agent. Make an interesting story. Glyphosate lesson. Glyphosate is a modification of the amino acid lysine, and it's the active ingredient in Roundup. It's real good at killing weeds. And what it does, it interferes with some enzymes that are necessary for plants to generate the amino acids that they need. We can ingest plants and animals and essentially steal their amino acids, but plants have to produce all of the amino acids that they use, except for the carnivorous flytrap. It takes some from flies. But for the most part, plants have to make all of their own amino acids. This chemical interferes with a couple of enzymes necessary for the production of a number of amino acids that we need, or that they need, and thus they die. The trick is they've modified the food plants, the wheat and whatnot, to use a different pathway, so they're unaffected by glyphosate. And humans don't use the pathway either, so the enzymes we have are not affected by this. But there's gut bacteria you have which is affected, so you're not totally off free. So you shouldn't be consuming Roundup. And I guess the lesson here is it's possible to have chemicals in an alien world that might interfere with the molecular process inside our cells. And there's no way to know what we're going to run up against. These are specific problems of a general problem, which really is xenobiotics. It's known as xenobiotics. Xeno is stranger in Greek. Xenobiotics are compounds that are strangers to our biosphere. PCBs are a good example of that. In the 40s, that was recognized as a wonderful chemical, very stable, non-combustible, non-flammable, good heat transfer characteristics, apparently very useful in paints. Wonderful chemical, had a lot of good properties, but if it got into the human food chain, bad things happened. Today, there's over 200,000 xenobiotics that have been developed in the last 100 years here on Earth, And you've heard some of the horror stories of what those have led to. But we have government agencies, looking at these things, and if they're harmful, they're in theory pulled off the market. So we have some protection against xenobiotics here. But if you're wandering around on an alien planet in an alien biosphere, again, it evolved along a completely different pathway, you're exposed to thousands, perhaps millions of xenobiotics. And the consequences, again, are somewhat unknown, but it kind of boils down to the question, how lucky do you feel? So an alien biosphere offers a lot of opportunities for bad things to happen. So we can learn a lot by studying alien biospheres. We can learn a lot by studying alien cell structures. That's one reason it's important to get into space and actually look for alien life. There's lots of lessons to be learned from that. We do have to address ethical and safety questions if we want to try to colonize an alien biosphere. Can we do it? Maybe yes, maybe no. But at least for those of us interested in trying to colonize distant stars and other planets, we're probably looking at space settlements and terraforming as the way we're going to operate. Okay, here's a nice image of a planet undergoing terraforming. Here's a nice image of a planet that has undergone terraforming. But is terraforming practical? Yes, but it will take a suitable planet. You've got to find one, hopefully one that's sterile. And if it's not sterile, you've got to deal with the ethical issues of sterilizing it, which, again, we're going to avoid. Colonists will need some place to live in the meantime. It requires the creation. Terraforming essentially is the transportation of the biosphere here on Earth to another planet. And that's the bacteria, the fungus, the insects, the animals, the fish, the plants. We might be able to leave some behind, like ticks and mosquitoes and leeches, but essentially all life is going to be transported to this new world. And we'll create our Earth too, rather than moving into one that someone else has created. So at the end of the day, at the end of the process, we'll have a place to live, but also all the life that we've evolved with will have a place to live. So that's the talk, and at this point, I guess I'm ready for any questions. Sir.
If the planet is sterile, wouldn't you assume that it would be hostile to life or to the formation of life, or not have an atmosphere, or have lost its atmosphere like Mars, mostly due to changes in magnetic activity, loss of a magnetosphere, things like that?
Yes, absolutely. It'll be very hostile to human beings. Any human that sort of walks out, very much like Mars. If you walk out on the surface of Mars, you'll last, about 30 seconds? So yeah, it is going to be very hostile. But the process of terraforming is to eliminate that hostility and create a Earth-type biosphere where people can walk outside just like here and exist just fine. Kent?
It takes a lot to transplant an ocean to another planet.
Well, you don't have to transplant the whole ocean. You just find a lot of water somewhere, and usually in the outer solar system there's lots of water, but you will have to transport life from Earth to colonize the ocean. Kent?
Do you think people who are saying we have to, essentially move to other planets, people like Stephen Hawking and Elon Musk, do you think they're aware of this kind of thing, possible incompatibilities of amino acids and things?
You would have to ask Stephen Hawking. I suspect he's aware of it, but just because there's a problem doesn't mean that there's not a solution. And I think terraforming is probably the solution that I would propose. There may be other ways to get around it, but it is an issue that needs to be, factored in.
Because I tend to think that the issue more often is how do we get there? The technology to get us to these places, and not so much, okay, what is it we're going to be dealing with when we get there?
Yeah, I've avoided the ethical question and I've avoided the transportation problem because both of those are, I think, pretty hard. They're in the back.
Assume a sterile planet 8,000 miles in diameter to give you one Earth normal gravity. What kind of energy budget do you project for terraforming that?
A lot more than we have now. But keep in mind, if we can transport, a massive world ship from here to there, we're dealing with the kind of energies that we need to terraform. So, can we do it today? Can we terraform Mars today? No, just because of the energy constraints. But you're exactly right. John.
I really appreciate everything you've said. To me, it underlines the importance of close examination of all of the planets and satellites in our own solar system. I mean, you've alluded to the fact that, well, not Mars itself may be sterile, and that's very important. But then we're already debating whether Enceladus and Europa may have life beneath the frozen surfaces and so forth. And hearkening back to when I was very young, I used to read John W. Campbell science fiction, and he assumed that since we already knew that the gas giants like Jupiter and Saturn, Uranus and Neptune were full of carbon compounds and an enormous range of conditions, that there may be life forms that are floating in the atmosphere at just the right levels. And so exploration of our own solar system and determining what's there and which of these processes have predominated is vital to the whole question of what happens when we get elsewhere.
Yeah, again, one of the problems with trying to put together a talk like this, is we don't have any data on what an alien life form or life system is going to look like. We don't know how it's going to be put together, and especially if you're looking at some of the, moons of the gas giants where they're going to live in an ocean covered with a, thick layer of ice. That's very alien to anything we understand. So I think it's imperative to get out there and look around and see what we can learn. And there's a lot we can probably learn in our own solar system. Even if Mars is sterile, it may have been that life evolved on it and then died out. It may be that life, evolved or, arose on Mars and then it was transported to Earth. There's one theory that we're Martians, that all life on Earth comes from Mars. So, getting out there and figuring out what's going on, I think, is imperative because, hey, in this universe, what you don't understand can really, really kill you. Okay.
Express one other opinion. For years and years, we always assumed that oxygen was the indication of life because we assumed that photosynthesis was essential. And then we discovered the worms under those rifts, and the fact that life certainly appears to be able to live in a completely alien environment there, and we would assume that that's inside of Europa and Enceladus. So you have to broaden the definition of life accordingly too, because it may be conceivable that something could exist in the observable atmosphere, which would still not be free oxygen.
Yeah, keep in mind, life existed on this planet for close to 1 billion years before it figured out how to do photosynthesis. So life can exist very comfortably, without an oxygen atmosphere. And yeah, your point's well taken. But if there's oxygen, I guess my contention is there's probably life on that planet somewhere pumping out oxygen. Okay, David?
It would be really nice to have an instrument that we could drop or in place on Europa or a distant Earth to Pluto to analyze for the presence of different types of amino acids, what they might be, what might be the distribution. Some distributions would suggest patterns of life.
Mm-hmm.
Have you seen any attempts at designing such an instrument? How big would it be? What would be it? It's like a mass spec maybe, That just tells us basics.
Yeah, basic elemental.
So how
Composition.
small, how compact, how simple can such an instrument be?
Well, again, remember the argument I made that nanotechnology is indeed possible. It exists inside our cells, so therefore I would think that you could build a fairly sophisticated instrument, very small, maybe the size of a dime, using nanotechnology. But nanotechnology has its own dangers, so yeah. Okay, gentleman here.
You hypothesized a sterile planet. The Earth presumably, in some sense, once was sterile, but it was colonized. What is the chance, do you think, that there will be a sterile planet which has already been colonized by the process which colonized us? Because that's been going on for as long as as long as the Earth has had life. So the chance of us getting there first strikes me as zero.
So that is one open question. How common is life throughout the universe?
No, but the means of transmission of life which brought it to us.
Mm-hmm. Okay, that sort of delves into the panspermia theory. You're familiar with that? The idea that life evolved one place in the universe and then spread out from there. So every life system we're going to run into is essentially based on the same set of blueprints.
That's
So,
like uniqueness of the sun.
yeah. Some very good questions. You asked some very good questions. I can't really answer them, again, because we don't have any real data to go on. It may be life is very, very common, that we won't find any sterile planets, in which case we may have to sterilize it and then proceed with the terraforming process. But hopefully we can find some naturally sterile planets, perhaps like Mars. Mars appears to be very sterile right now. Just working off that. Or, far-fetched, I guess you could actually begin to think in terms of building your own planet. It's interesting to note that, if you collided Venus and Mars together, you'd end up with a planet about the same mass as Earth. And you'd get rid of all that pesky CO2 on Venus as well. So, yeah, building planets is not too insane. Okay, David?
This is a weird question. I guess starting about a month ago, we had for the first time in all our written history, all our history, the realization that there's something from probably outside our solar system now passing through the solar system. And based on its velocity, it's easily would escape the solar system. So probably it came from a great distance. Have you seen any really credible way that we could use the technology that we already have in place to reach this asteroid or whatever it is that's passing through our solar system?
Oh yeah, I mean, we've got probes to Pluto and beyond, and I think the challenge there would be the velocity that this thing has as it zips through the solar system. But the Starshot people are talking about accelerating research craft up to 20% the speed of light. So if that's possible, we should be able to catch up with this thing, and circle it with probes, and the probes will then go with it. But yeah, the more we learn, I think the better off we are. Right now we're speculating in the dark, and that's an uncomfortable position to be in.
Let me come back to you a little bit. If you suggest that life originated in one place, but it reached us,
Mm-hmm.
therefore we are within its sphere of influence, if you like.
Mm-hmm.
We've got to overtake it to find a sterile place.
I don't think it came from anywhere except it originated here. I mean, it's just a matter of chemistry.
Well, the panspermia theory is just a theory, and it may be that life, once it originates somewhere, you know, will spread throughout maybe its solar system, you know, and
It only makes the problem worse. I mean, your hypothesis is find a sterile planet.
well, no, that's part of the solution. You know, first of all, find a sterile planet. I like sterile planets because you avoid the ethical issues of looking at a planet that is, you know, has life on it. And if the compatibility problem is real, the only solution is, you know, we live in space habitats or we sterilize the planet and then proceed from there.
I believe the discussion has gotten a little bit off the mark. Linda brought up the right point that the other alternative to panspermia is that life spontaneously occurs everywhere that you have the right raw materials and the right
Right.
energetics, so which seems more likely than panspermia, and it's in accord with the Miller-Urey experiment that you mentioned originally, the things that Cyril Panamparuma did after that, and so there's a very strong case for the creation of the proteins and stuff spontaneously.
Mhm. Yeah, that's a good point. Okay, well, thank you very much. Very good questions.